Okay, good morning all of you. So let's continue the discussion of this particular molecular biology questions. Okay, so friends, these are all the details about the venture batch 4, which is going to be commenced from the 26th January in biotechnology, chemistry, physics, as well as the mathematics. So on 30th January, All India IIT JAM mega mock test is going on, so which is absolutely free. Please kindly enroll in this particular test, and please stand a chance to win the Amazon vouchers worth of 65k. So to get a 10% of discount, please kindly use my code ChanduLive. So these are all the list of entrance exams, list of tests that are happening in the platform in the month of January. Full length mock test, T10 format, T20 format, mini mock test, and miscellaneous. Some are for the, what we can say that, plus students, and some are for the free I mean to say people can take it freely. Even some tests are exclusively for the plus subscribers only. So one mock test is going on, mega mock test is going on 30th January. Another mega mock test is going on 6th February. So please kindly make use of the two compulsory. Okay, so now let's try to understand. Okay, just a minute, let's start with the discussion of questions. Okay, I hope everyone is able to see the questions. I want every one of you think and answer very specifically. Okay, kindly don't be hurry. Think and answer. Okay, let's start. The question number 1. Holiday junction is observed during. Yes, everyone. Question number 1. Holiday junction is observed during. Anyone? Holiday junction is observed during. Okay, Prema, holiday junction, dear, it won't appear during the interphase actually. See, this is one homologous DNA. This is the another homologous DNA, right? So when strands are exchanged between the two DNA, you are able to see here there is a H-like junction, which we call it a holiday junction, and which will undergo the immigration also inner side. This holiday junction can be appeared during the process of recombination. Okay, it's not during the interphase. No problem, no problem. Okay. Question number 2. Type of proofreading activity by DNA polymerase 1 in E. coli is. Type of proofreading activity by DNA polymerase 1 in E. coli is. Correct. So whether it is a polymerase 1, polymerase 2, polymerase 3, generally can synthesize the new strand in 5' to 3', and they will do the proofreading in 3' to 5'. But we also know very well the DNA polymerase 1 can do the proofreading in 5' to 3' also. But they did not ask a specific activity, specific specific proofreading direction. They asked a generalized statement. Of course, the answer will be 3' to 5' only. Always remember that. Okay. Hence, the correct answer will be C. Good, good, perfect, perfect. Yes. Question number 3. This question, please think and answer. Reverse transcriptase of retrovirus is? Reverse transcriptase of retrovirus is? See, Prema, listen here. Anyway, most of the students will get confused about this particular statement actually. Reverse transcriptase. First of all, what the reverse transcriptase enzyme will do, it is going to synthesize the DNA. From where? RNA. Correct, na? So, we are involved in the synthesis of the DNA. So, DNA can be synthesized by which polymerase? The replication can be done by the DNA polymerase, there is no doubt about it. But this DNA polymerase depends on what? This DNA polymerase depends on the RNA. That's why we can call it as a RNA dependent DNA polymerase. So no dear, yes, very good. C will be the answer. Correct. Because we are synthesizing DNA, so DNA polymerase, we are synthesizing the DNA from the RNA, the instructions given by the RNA. That's why we can call it RNA dependent DNA polymerase. Very good. Yes. Next, question number 4. Amino acid sequence for a protein is known. We can estimate the sequence of mRNA coding that particular protein. It's a logical question actually. Try it. Try it, this question. No problem even though it's wrong. Try it. Okay, no problem. Look into this. Amino acid sequence for a protein is known. We can estimate the sequence of mRNA coding that particular protein. So what we have to understand, precisely to certain extent, if codon frequency is known. That is what exactly, friends, we will be calling it as what? Option C. Okay, that's okay, fine. That is actually a kind of logical question. Think and answer this question. Question number 5. Very logical. Regarding gene expression in prokaryotes and eukaryotes, which statement is correct? They are asking, friends, which is correct statement. Anyone? Regarding gene expression in prokaryotes and eukaryotes, friends, they have asked a question which is correct with respect to both. Anyone? Question number 5. Very good, Pechmuthu. Good. See, friends, because mRNA and DNA are not collinear. mRNA and protein synthesis simultaneously can occur. See, this is occur only in the prokaryotes. But you know, right, in case of a eukaryotic organism, mRNA and protein synthesis cannot occur simultaneously actually. Processing of hRNA yields mRNA. No. RNA polymerase can bind to the promoters situated on the upstream to the gene. Of course, true. See, this is what RNA starting site. So here what is there? Promoters are there. So exactly here RNA polymerase will come and attaches. Both in case of prokaryotic organisms also, even in the eukaryotic organisms also. Good. Hence the correct option will be, friends, D. Next, it's a general question, you have to remember it, but just try it. The percentage of protein coding sequence in E. coli genome is? The percentage of protein coding sequence in E. coli genome is? Okay, friends, even though you don't know the answer, you have to remember it. Okay, so this is 70%. Suppose all of you listen, this is the entire genome of an example E. coli bacteria. So this is total 100% of the genome. What is the statement I want you people to remember here? Out of 100%, up to 70% of the entire genome can code for the protein. Okay, good. Correct. Okay, think and answer. RNA dependent DNA polymerase is present in a human papilloma, hepatitis B, hepatitis C, Epstein-Barr virus. RNA dependent DNA polymerase is present in? Try it. Yes. See, friends, please remember RNA dependent DNA polymerase is present in the HPV, human papilloma virus. Please remember it. No problem. Yes, answer the question number 8. Semi-conservative model of DNA replication can be experimentally demonstrated by using? Semi-conservative mode of DNA replication can be experimentally demonstrated by using? Okay, so friends, fibromouracil, which is a kind of base analog. We will use majorly fibromouracil, which is a base analog for the DNA replication. So the correct answer will be A. Okay. Question number 9. If mutation in regulatory gene increases 1000-fold efficiency of repressor protein to operator under such condition, which is true about operon? Friends, this question is logical. You have to understand, repressor protein expression become more, very high fold of repressor expression. During the high fold of the repressor expression, what is going to happen? That's the question. Yes, what's the answer? Question number 9, what is the answer? Anyone? See, friends, look into the question carefully. If a mutation in a regulatory gene increases 1000-fold efficiency of repressor protein to operate under such condition, what's happening? Good, 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 good. Friends, see, all of you try to understand. This is what regulatory gene, which is gonna synthesize the repressor. Now, where this particular repressor will come and attaches, we all know that repressor will come and attaches to the operator. Now, if the repressor come and attaches to the operator, to the promoter, RNA polymerase will come and attaches. But do you think RNA polymerase can cross the operator? No, it never happens. If the RNA polymerase enzyme never cross the operator, undoubtedly there is a no protein synthesis. Correct, correct. Very good, Pachimuthu. Answer will be A. Good, 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 good. Question number 10. Try it, try it. Good, good. Okay, friends, no problem. Try to understand. Replicative transposons increase their copy number by replicating its copies and inserting into new position while keeping it copy at original place. The enzyme involved in this process. Anyway, if we want to move one transposable element from one location to the another location, what do we require? Of course, we require an enzyme transposase as well as the resolvase. Resolvase is the enzyme which is involved in the formation of a transposase co-integration complex. Okay, no dear. Okay, it is responsible for what? The formation of a transposase co-integration complex. For the transposase co-integration complex. For this particular transposase is required actually. No dear, no problem. Prema, please remember the option dear. It's a C. Okay, next. Inhibitor of RNA polymerase during the elongation of transcription in prokaryotes and eukaryotes both is. You have to choose an option of an antibiotic which can block the RNA polymerase enzyme in both prokaryotes also as well as eukaryotes also. Anyone? Question number 11. Okay, so friends, please listen everyone. Streptomycin is the broad spectrum antibiotic which can block the action of RNA polymerase during the process of elongation both in prokaryotes also as well as the eukaryotics also. No, rifampicin can only act on the prokaryotes, but streptomycin can act on both prokaryotes and eukaryotes. And actinomycin D also act on the eukaryotes. This is only on prokaryotes. Alpha amanitin is a fungal toxin. It acts on the polymerase enzyme. It won't affect the elongation process. It won't affect the elongation. The correct answer will be option C, streptomycin. Yes, question number 12. Option D here is IR light. No problem, no problem. They're asking friends, photolyase enzyme activate by absorbing which light? or our photolyase enzyme is going to repair the particular damage of a pyrimidine dimer by absorbing which light? That's the question. No, Prema. No. Dear, big damage only caused you because of UV light. How can we use once again UV light? See here, this is called as what? Pyrimidine dimer. Correct na? Whereas two pyrimidines associated with each other, especially thymines. This dimer we have to break. How can we break this particular dimer? By a specialized enzyme called as a photolyase enzyme. This photolyase enzyme, how basically it is going to show its action? Suppose imagine this is a photolyase enzyme. Photolyase enzyme having two chromophores. One we call it as FAD minus, another one we call it as a folate. You know what the FAD minus will do? It starts absorbing the visible light. In the visible region, which light it is going to absorb, friends? Of course, it is going to absorb the blue light. Whenever it absorbs the blue light, the point I want you people to remember here is that that particular blue light, after absorption of blue light, FAD minus get excited, it release the excited electron. That excited electron break this particular bridge. Later, FAD minus immediately get come out, as well as the enzyme also comes out. So the answer will be blue light. Okay, answer will be C. Okay, question number 13. Anyone? In E. coli, lac operon, see both positive regulation is they have given and negative regulation also they have given. What do you mean by that? That's a simple direct question. Okay. No dear, see Balu, they have given both positive regulation also, negative regulation also. See, negative regulation is basically what? Using of lactose, I mean to say. Negative regulation is actually in the absence of lactose. But they have given both positive regulation also. When there is a presence of glucose, what is happening? First the bacteria starts using the glucose. Once the glucose source is over, then they will come to what? Lactose. So which means that it can use both the glucose as well as the lactose simultaneously. If it is only negative regulation, then we can say only lactose. When there is a presence of glucose, then the positive regulation can be mediated. Hence the correct answer will be C. Okay, I hope it's clear for everyone till question number 13. Okay, so that's all for now. We will meet in the next class in the question discussion. Okay, friends, thank you all. I'll see you in the next class. Thank you all.